Dust looks dead, but it is in fact alive, teeming with hardy microbes traveling around the world, and somehow they're surviving the trip. How? Scientists think biofilm formation may play a key role. Welcome to Microbial Minutes, the American Society for Microbiology's update on what's hot in the microbial sciences. I'm Madeline Barron, Science Communication Specialist at ASM. Before we dive into today's video, please take a moment to fill out the survey linked in the description. It only takes a few minutes and helps us make better videos. We appreciate your support. It is estimated that between 500 million and 5 billion tons of dust is whipped up into the atmosphere every year. Much of this dust comes from deserts and arid lands like the Sahara and Gobi deserts in Africa and Asia, respectively. Dust storms in these areas spew clouds of dust into the air where it is then transported far and wide. Indeed, dust events in the Sahara can send dust all over the place from Europe to the southern US. Now, these dust particles traversing the globe may have tiny microbial passengers, including an array of bacteria and fungi. The composition of dust-associated microbial communities depends on different factors like temperature, humidity, geographic origin, land use, and wind currents. What is consistent is that dust is a vehicle by which microbes are transported to new locations and hosts. For example, dust can spread plant pathogens or alter the composition of marine microbial populations. It can also harbor organisms that cause disease in people and animals, such as Enterococcus bacteria and Aspergillus fungi. Desert dust in particular has been linked to increased morbidity and mortality due to respiratory disease, among other health conditions. Still, the exact health and environmental effects of dust-associated microbial communities are largely unclear and are active areas of investigation. What's also largely unclear is how exactly microbes survive the relatively hostile landscape that is a dust particle. Spore-forming bacteria, like those in the Bacillus genus, are known to be common members of the dust microbiome, with sporulation, that is, the ability to form spores, being a key persistent strategy. But as they fly through the air, are these bacteria active, or are they always dormant as spores? What are the mechanisms that shape their survival? Understanding these questions can provide more insight into the factors shaping dust microbiomes and their potential impacts. To that end, a recent study examined dust storm-borne Bacillus isolates to understand their growth patterns and dust colonization dynamics. The isolates were sourced from samples collected during spring and fall dust events and represented a broad repertoire of species. When grown under various culture conditions, all the isolates exhibited complex 3D morphology, indicative of biofilm formation under at least one condition. These findings suggested that for these dust-associated bacteria, biofilm formation, not just sporulation, could be a survival feature in the dust storm setting. The researchers found that the isolates exhibited biofilm formation under conditions reminiscent of those during a dust storm, such as higher temperatures and low humidity. They also found that growth of a representative B. subtilis strain grown on agar supplemented with sterile dust led to increased expression of biofilm development genes, plus more robust growth compared to bacteria cultured on agar without dust. When B. subtilis was incubated on agar containing sterilized dust from dust storms, which contains the residual metabolic output from original microbial colonizers, and self-made artificial dust, the bacteria grew better on the sterile dust. Mutant bacteria deficient in components of the extracellular matrix that encapsulates biofilms did not colonize as well as wild-type bacteria. In competitive fitness experiments, the mutant made up 30% of the population, while the wild-type strain made up 70% on sterilized dust. These results suggest two things. One, the organic component of dust is key for bacterial survival. Artificial dust lacking these residual components did not support robust growth. And two, the matrix proteins associated with biofilm formation are essential for colonization and survival on dust. These components are public goods that protect cells from external stressors and help trap and transport nutrients. They appear to be crucial to survival on dust. As a final step, the scientists used live dead staining on natural hydrated dust and observed rod-shaped cells forming distinct microaggregates on the dust particles. The authors claim these observations, quote, provide direct evidence of the functional significance of biofilm formation within the dust microbiome. So why does this matter? Well, it suggests that the extracellular matrix components of biofilms may protect against environmental stress and enable survival and attachment of bacteria during transport on dust. The study also indicates that through interactions with the metabolic output of other members of the community, Bacillus bacteria can successfully colonize nutrient-limited dust particles and remain active while doing so. That is, while dormancy via sporulation is one strategy for persisting in the harsh dust world, biofilm formation may be another. This study barely scratches the surface of all there is to learn about dust-associated microbes. There's plenty left to explore regarding the form and function of dust Bacillus biofilms and potentially those of other species as well. Diving deeper into their interactions and signaling of the dust microbiome in general is useful for understanding their impact on the environment on which dust particles land. Before we wrap up here, it's also worth highlighting that there is an active branch of dust microbial research focused on indoor or house dust, too, along with the outdoor stuff. We know there are health implications of house dust, which is shaped by the chemicals people use in their home, the people who live there, pets, and more. Dust and its associated microbes can be drummed up into the air during routine activities like walking and vacuuming or opening curtains. Research suggests that dust may promote spread of pathogens, including viruses like influenza A. As such, dust is more than just the stuff that collects on our bookshelves. It's a dynamic ecosystem that moves with us, shapes our environment, and may influence our health and that of the world around us. That's all for today. If you liked this video, subscribe to ASM's YouTube channel for more Microbial Minutes. As always, I want to thank you for listening, thank Ray Ortega for production, and I'll talk to you next time.